Can you talk,
Okay.
Okay.
So, welcome everybody. I have the pleasure to present Anna Protasio. She's an Uruguayan researcher. She studied biochemistry in the University of the Republic in Uruguay. She started working in first years in molecular biology by studying the antioxidant pathway of the tapeworm Echinococcus granulosus. It is during this work that she got a grant to go to the Sanger Institute, and she didn't come back because she went to do her PhD under the direction of Matt Berryman. She studied, during her PhD, she studied the transcriptomics of flat tapeworm Schistosoma ansoni, and afterwards she went to work on a postdoc where she studied non-coding RNAs. Specifically microRNAs that control genes in schistosoma also. After a second postdoc in India, she came back to the University of Cambridge, where she's a group leader I will tell you the exact title. She is a research group leader in computational biology in the Division of Microbiology and Parasitology of the University of Cambridge. She will tell her about her current work and interests. So I leave you, Anna.
Thank you, Christian. Thank you, everyone, here and online, for showing up for this seminar. I know it's an unusual time and an unusual day, so hopefully I can keep you entertained and keep you awake for the next 40 minutes. Well, I'll present to you the latest work that we've been trying to achieve in my team. It's a very small team. I've only started being a group leader 2.5 years ago, so this is all very new territory to me. And it was in collaboration with Christian and with Christoph Grunau that I sort of started this work on the non-coding genome and looking at subtelomeres and transposable elements particularly. Before I start, I'd like to thank Semed for inviting me to be here today, not just for the seminar, but I am here for the whole length of June. So if people find anything interesting about what I'm talking about today, I'll be more than happy to meet up some other time to talk about this, or this afternoon as well while I'm in Montpellier. So the title for this talk is Subtelomere Plasticity in Parasitic Worms Linked to Gene Expansion. And most of the work that I'm going to present today along this topic, it's a result of a PhD work of my current PhD student. So before diving into the hard science, I'd like to introduce, there's these two hats that I wear in my lab. We have two main areas of work. I traditionally been working on schistosomes for much longer than I like to accept, so nearly about 15 years now. And as Christian described, I started working on the protein coding aspect, looking at gene expression changes in life cycle stages of the parasite. But then very soon I realized that protein coding genes only occupy about 3% of the genome, 2% of the genome. So I started to think a little bit more about what the rest of the genome was doing there. And where we understand now in model organisms that the non-coding genome plays a very important role in regulation. And again, this is a bit of a dark matter. We still don't know what it's doing. So what I propose in my independent career is to understand more about the non-coding genome. And this leads undoubtedly to the study of evolution of genomes. It's through comparative genomics that we can reach to understand the meaning of these elements and what do they do. The other hat is a relatively new hat, and it's the one that looks at avian pathogens. And hopefully, before we finish today, I'll have a little bit of time to tell you a little bit more about this. This is something I'm very excited about. It's a new project, it's a new area. We work on understanding prevalence of pathogens in gastrointestinal tract of birds, but also in blood. I understand some people in Montpellier work on malaria and avian malaria. I'll be interested to talk to them about this. Yeah, and this is very topical now. There's infections and viruses coming in from the air. So hopefully we'll have a little bit of time for that. So this is the small team as of March 2023. And again, this is a mixture of computational, so dry lab and wet lab. I wear both hats, luckily. I'm able to jump from the bench to the computer seamlessly. And the work that I'm going to show today, I said, is from a PhD student. His name is Toby. It's the one standing right next to me. And when Toby arrived to the lab in October 2021, we were just coming out of the pandemic. We had been in front of our laptops for far too long. And we were excited to get new people in the lab and start to work collaboratively. During this time, I had been working with Christian well over a year by then. And we had what we thought it was a very well-finished library of transposable elements in the parasite that we're interested in. What are transposable elements? Transposable elements are pieces of DNA in the genome that can jump around and can therefore impact the evolution of these genomes. So there's a chapter of our story in collaboration with Christian that we're writing up at the moment, and that work is going to have its own kind of spin-off. But one of the things that I entrusted Toby to do was to look at what was the topography of those elements in the parasite that we work on. So before diving into the genomics, I'm also going to do a very short presentation of the parasite that we work on and why do we choose this parasite to work with. Schistosomes are perhaps somehow well known to some of you. For those who don't know them, these are rather complex animals. They are fully grown worms with their guts and the nervous system. So this is very far away from your unicellular pathogen like malaria or trypanosomes. They infect vertebrate hosts and also invertebrates. In this case, they are snails. And they have this circulation between a vertebrate host and the snail. And in the meantime, in the middle, have a plethora of different we call them life cycle stages. So the parasite changes from one form into the next. Characteristically of this class of worms, they have two sexes. So most of the worms that belong to the same family are hermaphrodite. These guys have two sexes. You have a male and a female. And here on the right-hand side, you can see this is a pair of worms. The male is the chunky one that is at the kind of on the outside, and the female, you can hardly see it. You can see the head here peeping out at the top, and then the tail at the bottom. And the female is contained within the male, and this is how they copulate. So it's almost like when you see them together, you can see that they're hermaphrodite, but they are not. You can pull them apart. And they have this amazing phenotypic plasticity that depends on proximity of the male for the female to be reproductively active. Now, for the purpose of this talk, I'm going to bring up one very fantastic aspect of schistosomes, which is that the worms cannot be detected by the immune system. The worms they live in the circulatory system. Depending on the species of schistosome, they could be next to the guts or next to the bladder. And they are not attacked by the immune system in any way. Worms can live up to 30 years inside a host without even blinking. So they don't cause pathology per se. What causes the pathology is the egg of the parasite. And that's the beast that you see at the top of the worms. So that's about 200 microns in length. The females produce eggs continuously, depending on the species in terms of the amounts. And what happens is that when the worms are in circulation, those eggs get flashed back into the liver if you have intestinal schistosomiasis and the worms are near your intestine. These worms get lodged these eggs get lodged in the liver, and it's the immune response against the egg that causes the disease. So it causes accumulation of immune cells around the egg and immunopathology. So people who have severe schistosomiasis will have liver failure as one of the consequences of this. So going back to let's talk about a little bit about the genomics. So when I was at the Sanger Institute, when I arrived there in 2007 to do my PhD, my boss, Matt Berryman, was kind of finishing the first, what we looked like a good draft at the time. There was 19,000 pieces. Now we have them in 8. So it was a long way to walk between then there and now. And I was very happy to be part of that journey. Mansonite in particular, which is the worm that we're going to talk about today, is particularly amenable to laboratory work. So we can keep it in the lab very easily, which means that it's perhaps the model organism for all this class of worms. So we have a pretty good assembly, a pretty good genome assembly. What this that reflects we have a reference genome of this parasite. So when Toby arrived to the lab, he was interested in understanding more about the dynamics of transposable elements. And we had this library of transposable elements. And the first task for him was like, okay, well, go to the genome and try to find out where these transposable elements are. Transposable elements are highly repetitive. The genome of schistosome is about 35 to 40% repetitive, which is the similar repetitiveness that we have in our genomes. So this is the first plot. I'm afraid it's a little bit squashed there on the slide. I think here, perhaps at home, you can see it better. Hopefully, you can appreciate this is a circus plot. And what we have here represented in the outer ring, we have the frequency of transposable elements, these repeats. In the middle, we have something called W repeats, which are very specific for schistosomes. In fact, very specific for schistosoma mansonite, and they're accumulating one of the sex chromosomes. And in the inner ring, you have GC content. And one of the first observations that we had was that the frequency of transposable elements would increase towards the end of the chromosome. We were very lucky that we have an assembly that is almost telomere to telomere. So we know that in some of these chromosomes, we can reach up to a telomeric repeat. So we were quite curious to understand more about what was going on in there, in those regions. So let's refresh a little bit about how we understand chromosomes and what are the overall kind of bird view structure of chromosomes. So this is a representative and a schematic. And we're going to talk about telomeric repeats, and within metazoans, most metazoans, this is a very conserved hexamer that has a repeat TTA, GGG, and we expect when we get to the end of those chromosomes to find those repeats. Most of the genes are in the chromosomal body, and this is kind of the important protein coding functional part of the chromosomes. And then there's this very difficult to define subtelomeric region, which comes somewhere between the telomere and the chromosomal body. And if you go out, I mean, we've been talking about subtelomeres as a research community for many decades, but if you go and you try to find a definition, you're not going to find a uniform definition of subtelomere. In some organisms, they'll say that the subtelomere is from the telomeric repeats to the first housekeeping gene. In others, they will tell you it's the first million bases. None of these definitions sort of worked for what we wanted to do in schistosomes, so we needed to find our own. The reason why we needed to define these subtelomeres is because we were very interested in trying to find out what's in them. What we could see from the transposable element frequency was that these could potentially divide the chromosome into differential topologies within the same piece of DNA. And different topologies will give rise to differential function. And what we know from other parasites in particular is that there's a lot of very important genes that are found at the end of the chromosomes. The end of the chromosomes in other parasites like in Plasmodium falciparum, they are quite likely to recombine, and they are hosting an array of different genes that have to do with immune evasion. So when we're starting to see some resemblance of what the chromosomes look like in Mansonite with respect to other parasites, we got very excited and we decided to investigate further. So, and this figure is just to exemplify the situation, what happens in falciparum. You have you have a schematic representation of the chromosomes from 1 to 14, and you can see that at the end of those chromosomes, there is an array of these genes called VAR genes that are the ones that encode proteins that are going to be expressed in the surface of the parasite and that help the parasite evade the immune response of the host. So pretty critical genes. We call them parasite contingency genes because they make the parasite lifestyle happen. So on the basis of these observations in other organisms and what we were seeing in schistosomes, we decided to investigate further. And the questions that we wanted to ask were whether we could find subtelomeric-like regions in subtelomeres. We kind of understand that perhaps a few hints based on repeat content were already there. Whether these regions in schistosomes were also likely to recombine, and whether these regions, or what sort of genes were confined in these regions, if any, because it could be that there are no genes at all. So we set off to that, and the first task that Toby wanted to do was to define these subtelomeric regions. And because we don't have a proper definition for it, we decided to do it based on repeat content. So what you can see in this plot, it's an enrichment estimation of how repetitive a given sequence of DNA is, what is the proportion of repetitiveness that we have. So you can take 1,000 base per window, and you can calculate what proportion of that is repetitive, and whether that is enriched in that particular part of the genome, or it is the same frequency that you would find somewhere else. And based on those calculations, you can calculate a p-value, you can put a cutoff, and you can say, well, these regions are highly enriched for repeat content. We know the subtelomerics in other organisms are repeat rich. So with this calculation, we were able to define these subtelomeric regions, and we could find them in all chromosomes except one arm of chromosome 5 that we believe is not complete. So we believe that it's not reaching the end of the chromosome. So now we have relatively well-defined what we think are subtelomeric-like regions. And the next step was to test whether indeed they are enriched for repeats. And within repeats, we have several types of repeats. We have simple repeats, which are a bit like microsatellites, some of you might be familiar with. We also have transposable elements, and we also have these W elements, which are very specific for Manzanite. And what we can see here is that in particular in these regions that we define as subtelomeric-like, there is a significant enrichment for transposable elements. So again, it's a little bit of a catch-22. We're just confirming that this is the case in our windows because we use repeat content to define them. Another characteristic, so now we define these regions that are highly repetitive. We think that they are subtelomeric-like. And the next thing we wanted to do was to try to find whether these regions were also likely to recombine in the same way that we see for other organisms, and in particular for the parasites. So Toby used a tool called BISER, and BISER is a bioinformatics tool that takes that looks at the genome as a whole, and it looks for areas of the genome where there is high level of homology on very large chunks. So we're looking at very large duplications or replication spots. And after filtering for repeat content, so we're taking out everything that looks like a transposable element, which we expect to see everywhere in the genome. We filtered out for things that are very small, so each of these data points are at least 20,000 base pairs. We can end up with something like this, in which each of these orange lines, and there are many in there, are linking two areas, two windows in the genome that are highly homologous to each other. And therefore, they are likely to have originated by duplication. As you can see, probably you can guess, these things are at the tips of the chromosomes. So on the light gray areas, over here and there, are what we previously defined as subtelomeres for all the chromosomes 1 to 7 and the sex chromosome bits. So we observe that perhaps there is an enrichment for these homology blocks within the end of the subtelomeres. We can verify that statistically by showing that that is the case, by counting how many of these windows are hit with the subtelomere. We compare how we would do at random. So at this point, what we have is we have a genome assembly which looks very much like a canonical structure with high repeat content towards the end of the chromosomes, where we also have these areas that are very rich in transposable elements and they are prone to recombination. So it's starting to look like a place where some regulatory activity could happen, that some perhaps gene duplication could happen as well. We can also confirm that, as expected for the definition of subtelomeres, we also have a depletion of genes in these areas. So there's a lot less gene density in the subtelomeres than what there would be in any other part of the genome. So the genes that ended up here might be quite they might be quite different from the rest of the genome. And which genes are here? I'm sorry, this is extremely small. I'm expecting slightly bigger screen. But we extracted these genes, which were not annotated. So we were very lucky to work with the people who assembled the last genome, and we asked them for help. And they very kindly obliged. And they annotated a lot on their own effort to try to annotate the genome in a better level. They also focus on those regions of the subtelomeres for which we knew, based on RNA-seq data, on transcriptome data, we knew that there was quite a lot of transcriptional activity, but there was no gene annotated in there. And give and take, I think there's about 200 new genes in these areas, from which 150 we were able to cluster into these sequence homology networks. So Toby went out, and BLAST was not good enough for this kind of thing, but he found this tool which looks at all the sequences together and they just cluster them based on sequence similarity. And now we can start defining, we were not expecting to find, very large families of genes that are very similar to each other and seem to have been expanded in the subtelomeric regions. So the dots in orange are genes that are in subtelomeres. The ones in blue are genes that are in the chromosomal body. Some families are mixed, like the one at the top left, that is an hypothetical protein family. So those of you who work on parasites know that about 30–40% of the transcriptome of the proteome of a parasite is poorly characterized, so we don't have the tools to know what these proteins do. But I'll tell you a little bit more about this particular family because they seem to be secreted, so there might be some role in the host-parasite interaction. Next to it on the right, there is a family of circadial proteases. These ones are all chromosomal. They're all in the chromosomal body, and probably they're undergoing a different type of duplication. We think that is a tandem duplication that they do. And some of the other ones are a mix of the two. In the case of the hypothetical proteins, we are particularly interested in, and we're planning to do further down more experimental work on this, depending on grants being successful. Because we believe that these have all the signals required for either secretion into the environment, secretion outside the cell, or for being anchored at the surface of the cell via transmembrane domains. We know very little about these proteins, but we know that they are extremely variable. There are 49 members of this. They're all located at the end of the chromosomes, and they resemble very much that structure of the bar genes in Plasmodium falciparum, or the VSGs as well in trypanosomes, which also have a similar architecture. Whether they undergo switching for different expression, we don't really know. What the expression data that we have looked at, we think is mainly expressed in the snail and in the snail stages and in the mutacidia stages, which is the stage that invades the snail. So perhaps they have something to do with the immune response in the snail itself rather than in the intramammalian host. So this is where we are with these projects. Toby is writing up this paper as we speak. And I guess what we can conclude from here is that schistosomes, now that we have the full assembly, we can understand that the topology of the chromosomes are perhaps a little bit more complex than we thought before. There is a very strong correlation between repetitive sequences and recombination events. This is not new in biology, but it's new for our parasites. And now we understand that it is in these regions which have been previously overlooked because repetitive regions were not able to be sequenced in previous genome assemblies efforts. We now understand that there's a lot more genes there that previously found, and that these genes are likely to change quite quickly, or they're able to recombine and reshuffle their structures, perhaps to accommodate some sort of fast evolution, fast adaptation that this parasite needs as it jumps from one host to the next, or from one environment to the next. So I guess at this point, I'd like to thank the people who helped on this particular work. And these are our collaborators, Steve and Matt. who are in Glasgow and Cambridge, Christian here in Perpignan, and Toby, who is the person who did all of the work. My funding is from Christ College, Cambridge, and we are hosted at the Department of Pathology. So this is the end of the first half of the talk. And I think, is it possible to take questions before we jump to the second part?
Yes, it's possible, but I couldn't say to people on Zoom.
Does anybody have any question here? Yeah. Yeah. No, no, no. So each chromosome, no, it's not circular, it's not bacteria, it's a metazoan. So each chromosome is independent. And yeah, so this is all chromosome 1, and that's the end of chromosome 1, and that's the end of chromosome 1, left and right, and this is the, yeah. Sorry, I should have said that.
Bruno on Zoom for some more information about the MEGs and the MERCs.
Right, okay, yeah, so he spotted the MEGs. So yeah, so there are 2 microexon genes that we find in the subtelomeres. I think there are 2 copies of MEG17 that you can see there on the slide. The mucins, so the mucins, oh yeah, this one, that's the 2 MEGs. So microexon genes, MEGs are something that only people that work on schistosomes know. And these are these very quirky genes that have exons as big as 3 bases. And we think that there could be some alternative splicing events that kind of jump exons, and that creates different moiety on the transcript. So yeah, there is a specific interest on these ones. So those are the MEGs, and then the mucins, we think they are in the chromosomal body, although we need to confirm that because the circular proteases are very well into the chromosomal body, but a lot of these families are kind of on the border. What we understand is a subtelomere. So if we are a little bit more permissive in terms of our p-value when we define the subtelomeres, maybe these ones kind of end up being in a repeat-rich region, rather than, so they could be that they're also recombining. There's clearly 5 copies of them in there. I hope that answered the question.
Thank you. I have another question on Zoom from Ronaldo Augusto. Are there any particular epigenetic feature in genes located in your telomere?
We don't know. The answer is that we don't know yet. That is work that needs to be done. Understanding what are the different histone modifications in those regions would be very interesting. And it's something that I'm sure the GRUNOW team can help with. I think that I can read the next one. So there is somebody's asking about, I think I'm not missing. Is there any recombination map available for schistosomes, and how is meiotic recombination distributed along chromosomes? That's a very good question. I understand someone in Perpignan is working on this, so perhaps they are in a much better position to answer this question directly. Yeah, so that's all I'm going to say. Yes, hello. Yeah, so the question is, I'll repeat for everyone else. The question is, there is higher frequency in chromosome 3 in the middle of the chromosome compared to others. That's very well spotted. You mean in here? This one? Oh, okay. So not this figure then. This one? This bit? Yeah. So I know that the ribosomal DNA genes are there. So maybe that's why there is higher repeat content there, because they will be identified as repeats. So yeah, we didn't look into that particular peak, but it could be attributed to ribosomal DNA genes. Yeah, they are in chromosome 3. Yeah, thanks. That's a very good observation. I think if there are no any more questions here or online. Okay, I'm gonna now switch, and I'm gonna do a very quick advertisement of the other project that we work on, in case people are interested. And this is something that I call poopomics. And yeah, is that good on Zoom? Okay, good. Right, so this is my little pet project that has taken over my life in a bit. So I love worms and so on, and this is how I ended up here, and I'll tell you the story in a bit. So yeah, so this is how everything started, and I don't get tired of showing this video. I'm really sorry the resolution is so bad here, but those at home will be able to see that that is a piece of poop from a bird. It landed on my trousers when I was in the park, and I was trying to take it off my trousers, and it had worms in it, and it blew my mind. And anybody that has chickens would be like, duh, there are worms in the poop, or anybody that has seen any birds before. But for me, who grew up in a city, this was mind blowing. And it will kind of open a new chapter into thinking about birds as a way of transmitting pathogens rather than just looking at like pretty things that fly in the sky. Because birds were here before planes were here. So when we think about how COVID moved around the world, we know that we did it getting into planes because we want to be places and we want to see people. But even before humans were on Earth, birds were already circulating pathogens around the world. I kind of knew a little bit about flyways, but I got really very interested in the interconnectivity of these flyways. So migrating birds, there's pretty much, in terms of movement, there's two types. There's the migrating ones and the resident ones. Some species can switch sometimes, some populations can switch from being migrating into being resident. But if you look at particularly at the migrating birds, you can see in this map, there's about 7 to 8 flyways. They call them flyways. So this is like routes where the birds will go along the length of the year. And they are also well interconnected. So it tells you that if a new pathogen jumps on these birds, the likelihood of this to spread very quickly around the world is probably 1 to 2 years. Because if it jumps from bird to bird, then it can easily just then jump from flyway to flyway. So with that in mind, and having seen the poop with the worms, we start thinking about what else is flying in these birds. So I started a project working on this. Pretty much the piggybacking on what we understand now is the current pandemic, the current outbreak of avian influenza. So as I was writing these grants back in August 2021, we got the first outbreak, the first report of avian influenza arriving in the UK, and that was very close to where I live. That was in Norfolk, and that is just a few kilometers north from Cambridge. So it was very timely as well to have a project in which we were using bird feces to understand more about pathogens in birds. So, I'm just going to tell you what kind of projects we do at the moment, and we're interested in all pathogens that we can identify. As long as they have DNA or RNA and they are in the poop, we can probably find them because we use sequencing technologies for that. We use molecular detection, but we also use shotgun sequencing. And what we want to define here is we want to be able to use methods that are non-invasive to the birds. So a lot of the methods out there will use cloacal swabs. We think that's quite invasive when you want to do it in a population. So we focus on non-invasive methods. It needs to be sample collection, needs to be fast. We also work on live birds. So a lot of the surveillance planning, the other surveillance that exists in places like the UK will be based on dead birds. And a bird that is dead is not going to be able to transmit the disease any further. So we're interested in live birds. And what we are interested in finding is pathogen prevalence. And we also want to, in the long term, be able to find hotspots of transmission, places where the birds are congregating and passing on the disease to other birds. And we also want to understand what is the fitness cost of these pathogens in the birds when they have them. And one of the, as an example, the study site that we use is this place called the Wash. The Wash is a very large bay in the east of England, and it's mud floods. So it's a very tidal area. When the tide comes in, the birds are roosting very far into the water. When the tide comes in, well, the birds fly up and they will start looking for new places to roost, that is to land and to kind of eat and do their thing. And we can take this opportunity to try to capture these birds and get our samples. I don't work along with this. I work alongside an association called a group called the Wash and Wader Research Group. They have about 60 years of experience working with waders, which are the birds that live in the shores. And this work would have been impossible without them. So this is a picture of us we're waiting for the birds to come down from they've been already exiled from far away in the sea. And you can see here the water is sort of rising. And we're waiting there for them to come, and there's a net we throw into them to capture them. One of the other methods that we use is mist netting. So these are nets that you put high, like a bit like volleyball nets, but they are much, they go almost all the way down to the water. We wait until the night comes, we put a collar. So these are speakers with the call of the birds, and the birds will come and try to find their friends, and they get caught in the net, and we can collect the birds. What is very good about working with this team, they have all their expertise, is that apart from getting samples, we can also have all the metadata of the birds. So we can know how much they weigh, we know the length of the wing, we can have a pretty general view of the fitness of the bird. So when we characterize the pathogens that these birds have, we can then also associate that with whether there is a particular cost or anything that tell us that this bird is ill or not as fit as it should be. And so how do we do the collection? In collaboration with this team, we came up with what we call bird toilets. And these are boxes that are shown here on the top left, that they have individual compartments with lids. And we put a cardboard box on the bottom. So when they bring the bird from the net, we put each bird in one of these boxes, we assign them a number, and we let them there for 15-20 minutes. They are very generous with their samples, so the birds poop a lot. And after that, we give the bird back to the bird bringers, and then they take it away and they will put a ring on the bird. So that bird will be forever matched with a number that we can then track back. And then all the metadata gets recorded. And we have then a match, each match sample with the bird. And during August 2022 and September 2022, we collected about nearly 500 samples using this method. And we are now in the process of processing these samples. So we are extracting DNA, we are extracting RNA, we're looking for viruses. We are trying to make libraries to send for 16S, trying to understand the bacterial population, trying to understand the parasite population by 18S and 28S. And I hope to be able to come back sometime in the future to tell you how the story ends. But because it's a very short project, a very new project, this is as much as we have at the moment. And this work has mainly been contributed. A lot of graduate students that were in the lab at the time, that spent the summer with us, came to us fieldwork. And also my research assistant, Kili, who's been working on this, and she's also a bird bringer. And that is it. So I think I am spot on time. Thank you very much. And I'll take questions for this part of the second part of the presentation. Thanks. I will, yes.
So in the Pupomics,
Yeah.
you have the sequence you say you want to try any kind of pathogens. So I guess viruses, bacteria, proteins.
Yeah, so the question is about the range of pathogens that we want or we are willing to investigate using the Pupomics approach. So because of cost constraints, my main focus is on 16S and a combination of 18 and 28S, by which you can get to the species level. So having a microbiome profiling and what I call like a parasitome profiling. These primers for 18 and 28S have been designed to kind of capture parasites mainly. They avoid the vertebrate host. And if people were to be interested in viruses, that's not something I'm looking to do right now, but the samples are there. We have 500 samples from this project only, but we also have another 2,000 samples from this last year in the freezer that have been collected throughout the UK by our collaborators. And the main point of that is just to do RNA extraction, look for avian influenza, and trying to set up this as a method for surveillance, a method that could be used for surveillance of avian influenza as an example, but it could be also other zoonotic diseases.
For the bacteria, for the 16S,
Yeah.
are you trying to identify potential pathogens, or is just to an idea of the microbiome?
Yeah, so the question was about whether we're trying to find pathogens based on 16S. So my understanding, I'm not a microbiome person. My understanding is 16S only, I will not be able to get to the bottom of it. So this is a first pass. There is the opportunity to do something like long read 16S, do using nanopore, which will probably get us a bit closer to a species level and be able to identify pathogenic from non-pathogenic. In the case, for example, Salmonella, I know there is different strains that can be pathogenic and non-pathogenic. Yeah.
I will try to open the microphone, so we start with
Okay,
Delphine,
sure.
if it doesn't work. Okay, so Delphine, if you are mute, you can try to ask a question.
Hello, can you
I
hear
think
me?
this one
I'm
is
sorry, I
off,
don't hear
that's
you.
the problem.
Yes. So if I,
Yeah, but then when I talk, it will echo.
sorry.
I think we're going to try to read your question, Delphine. I'm sorry. I'll read it for everyone. So how do you make sure what species the poop are from? That's a very good question. So there is on the first collection that we did in February and March 2022, we did environmental samples. And for that, we don't know the species, but we know that there will be some host cells in that poop. So we could potentially try to do mitochondrial sequencing. And with that, we could be able to identify the species. On the example that I showed today on the slides, we know exactly what those birds are because we can catch them. And the people that catch birds know a lot about birds, and we know exactly which birds they are. What we don't know sometimes is the sex and the age. We can estimate the age. I hope that answers your question. Then Christoph is asking how to make the difference between DNA from living pathogens that can be transmitted and dead parasites that are digested. That's a good question. I never thought about it like that. I wonder whether the digested DNA will be very, very small. No, I don't know. I'll take feedback on that. Thank you. That's a good question. I didn't think about that. And then Delphine is asking again, do you study different bird species? Are they all migratory? So we have samples from a lot. We have, so the project from the Wash is particularly migratory species because that's a hotspot for migration. We have red shank, oyster catchers, sanderling, a bunch of them. We also have resident species of passerines. So we have a collection of tits from the Oxford area that one of my collaborators, Michelle, sent us quite a lot. We also have curlew. We have lots of geese samples. I can give you, if you're interested in a specific bird, I can give you the species that we have. And if you want, we can collaborate on that, no problem. So we have resident and migratory species, I guess that's the answer. Okay.
We have a question on the chat.
Right.
Mohammed, can you see
it? Yes. So I'll get to it. So Mohammed is asking, how much is sufficient the adapted phenotypic plasticity and nematodes that is driven by variation in selection pressures across environmental gradients to sustain the survival of reproduction? I suspect this is referred to the previous presentation. And I'm going to read it again because I don't think I understood. How much is sufficient the adaptive phenotypic plasticity of nematodes that is driven by variation in selection pressures across environmental gradients. I don't know the answer to this. I'm not sure it has a lot to do with what I was preparing, but I will look into it because it's clearly relevant. Thank you, Mohammed, for this question. I will copy it and maybe I'll get back to you later if you can drop me an email and I can try to answer this question. Okay, I think that's it. Yeah, sure. So yeah, so when we do shotgun sequencing, when we have more money for it, we will be able to pick up what they're eating. Yes, you can already see in some of the birds because there's a lot of crustaceans. A lot of them have a lot of grass. There is a problem there with the extraction of DNA. I know chloroplasm will be kind of an invading DNA species, so we need to see how we're going to enrich. But yeah, it would be great to see that, especially there's some species we don't know what they eat. In particular, there are a few curlew. Curlew are birds that are about 30, 40 centimeters long. They have a beak that is bent, and they are a highly conservation concern in the UK. And there has been a new group of curlew that seems to be going inland. And so they typically feed on the shores, but we don't know what these, and there's people from the Washerway, the group working on this, they don't know what they eat because they go inland and normally that would not be their natural habitat. So it will be interesting to sequence the sample from those birds if we can find them to see what are the different diets between those ones that go inland with the ones that are on the shore. So that, yeah, that is one of the ways we can apply that.
Because you can link the parasite
Yes.
position with the diet.
Yeah, that could be. Yeah. There's one more. Okay. Yes.
Would you also need the sexual dimorphism within the class? What species?
We don't, so the question was whether we're going to be looking at differences in pathogens between males and female birds. I didn't think about it. I guess if we can sex them correctly, either via molecular tools or some birds are more phenotypically easy to do and others are not. I think that would be a good one. There's definitely going to be a column in our model if we have that data. Yes. Okay,
So,
excellent. Thank you very much. Thank you very much, everyone online, and I'll just stop sharing and close.